So let's add another example of how people are trying to alter plant properties at various levels. Genetically modified organisms, GMOs, have already been quite controversial, and people are always debating about whether they are healthy, they are safe, they are nutritional, and so on and so forth. So here is an example of tinkering with rice to boost grain yields while also shortening the duration of rice. This just came out in Science, and I thought I'll include it following the soybean photosynthesis paper we just discussed briefly in the previous podcast. The abstract here goes: complex biological processes such as plant growth and development are often under the control of transcription factors that regulate the expression of large sets of genes and activate subordinate transcription factors in a cascade-like fashion. So there are genes for certain functions, but their expression and the related genetic functioning together are controlled by these transcriptors. So this is talking about those transcriptors and whether those transcription factors can be modified to change the growth rate and yields. Here, by screening candidate photosynthesis related transcription factors in rice, we identified a DREB, dehydration responsive element binding, family member, OSDREB1C, I'll just say it like that, in which expression is induced by both light and low nitrogen status. So the signal is received that there is not enough light or that nitrogen is low, then this transcription factor kicks in and controls the gene expression. We show that OSDREB1C drives functionally diverse transcriptional programs determining photosynthetic capacity, nitrogen utilization, and flowering time. Field trials of OSDREB1C overexpressing rice revealed yield increases of 41.3 to 68.3%, and in addition shortened growth duration, improved nitrogen use efficiency. So when there is nitrogen limitation assessed by the plant, it may be because it's functioning at a very low nitrogen utilization efficiency, but nitrogen may be enough if its efficiency can be boosted, which is what is being done here. And this promotes efficient resource allocation, thus providing a strategy toward achieving much needed increases in agricultural productivity. Rice, of course, is consumed by billions of people, especially in Asia, but now across the world. And there are other debates about how to grow rice. Rice paddies obviously emit a lot of methane and so on. Can feel some wind. Hopefully the recording is not contaminated too much. So the introduction of the editorial summary of the article provides a nice way to understand this a little bit more detail. Rapid population growth, rising meat consumption, and the expanding use of crops for non-food and non-feed purposes increases the pressure on global food production. At the same time, the excessive use of nitrogen fertilizer to enhance agricultural production poses serious threats to both human health and the environment. So we talk a lot about carbon cycle, but nitrogen cycle has been grossly mismanaged by humanity, and it has had significant impacts on the environment, biodiversity, vegetation, coastal ecosystems, and so on. To achieve the required yield increases and make agriculture more sustainable, intensified breeding and genetic engineering efforts are needed to obtain new crop varieties with higher photosynthetic capacity and improved nitrogen use efficiency, or NUE. However, progress has been slow, largely due to the limited knowledge about regulator genes that potentially can coordinately optimize carbon assimilation and nitrogen utilization, which is basically a way of increasing yields. The rationale for this work is that transcription factors control diverse biological processes by binding to the promoters or intragenic regions of target genes, and a number of transcription factors have been identified that control carbon fixation and nitrogen assimilation. Obviously there's a lot of biology involved there that we are just plowing through. A previous comparative analysis of maize and rice leaf transcriptomes and metabolomes revealed a set of 118 candidate transcription factors that may act as regulators of C4 photosynthesis. You remember the C3, C4 pathways of photosynthesis that respond differently to temperature and CO2 changes, which is what we are doing now in global warming. We screened these transcription factors for the responsiveness to light and nitrogen supply in rice and found that the gene dehydration responsive element binding protein 1C, OSDREB1C, a member of the apetala-2 ethylene responsive elements binding factor AP2ERF. I'm just reading it. We don't know what all this is, but we just want to get a sense of how these things work. Exhibits properties expected of a regulator that can simultaneously modulate photosynthesis and nitrogen utilization. Obviously, these grasses have evolved to sense environmental factors that are important and modified their ability to grow, when to flower, and so on, so that resource use is managed, especially during limitation times, whether it's light or nitrogen. The conclusion of the study, I'm jumping the results part. Overexpression of OSDREB1C not only boosts grain yields but also confers higher nitrogen utilization efficiency and early flowering. So we are basically changing its phenology here and its function. Our work demonstrates that by genetically modulating the expression of a single transcriptional regulator gene, substantial yield increases can be achieved while the growth duration of the crop is shortened. The existing natural allelic variation in OSDREB1C, the highly conserved function of the transcription factor in seed plants, and the ease with which its expression can be altered by genetic engineering suggests that this gene could be the target of future crop improvement and more sustainable strategies towards food production. I think many of you know what allele is. You inherit genes from both parents, so crop is getting from male and female parents' genes. So you have a pair of genes and they are called alleles. The one that gets expressed is the gene and the other one remains unexpressed, right? So here is the summary figure. Transcription factor related to C4 photosynthesis, light and low nitrogen are sensed by this transcription gene, and they are manipulating the transcriptor, its expression, its control on the cascade of gene expressions that control these things into early flowering, enhanced photosynthesis, and improved nitrogen use efficiency to increase the rice yield by 41.3 to 68.3%, and increase nitrogen use efficiency, which means even when there is low nitrogen, it can still have enhanced photosynthesis and give you increased yields, which should drive reduced nitrogen use, which is what would be very good co-benefit as well. And the wheat yield, by the same method, wheat yields were increased by 17.2 to 22.6%. So carbon assimilated by photosynthesis increases when you enhance photosynthesis, and nitrogen is taken up by the roots, which is also enhanced by enhanced nitrogen use efficiency. Okay, so OSDREB1C coordinates yield and growth duration. OSDREB1C was identified by its responsiveness to light and low nitrogen in a screen of 118 transcription factors related to C4 photosynthesis. Transcriptional activation of multiple downstream target genes by OSDREB1C confers enhanced photosynthesis, improved nitrogen utilization, and early flowering. Together, the activated genes caused substantial yield increases in rice and wheat. Okay, good news. Is this a Frankenstein experiment? Not necessarily. These people are hopefully very careful, but also with genetically modified organisms and the seeds that are more drought resilience, high yield, high temperature resilience, etc., have been causing some issues in terms of who owns the seeds. So for example, farmers may buy the seeds, but they cannot save the seed for next year. They buy it once and then they have to buy it again next year. So all kinds of details are there in terms of who controls these intellectual properties and ownership of patented seeds, and how farmers are being made to pay for those things. So those issues will hopefully get resolved, but this kind of science which advances genetic engineering of early flowering, increased yields, enhanced photosynthetic efficiency, and so on, obviously will be very useful as populations will grow for some more time, and then crop yield demands, crop food security, crop grain demands will go up, and so on. Okay.